Drop water onto what looks like a dead, crumpled speck of dust, and something impossible can happen. The speck swells, unfolds 8 tiny legs, and walks away. This is a tardigrade, and no, it is not immortal. It cannot casually swim through boiling water, live forever in space, or survive every disaster. Its real ability is stranger. When its watery home disappears, some tardigrades can temporarily shut down almost everything that makes an animal seem alive. To understand that trick, first look at the active animal. A tardigrade is usually less than a millimeter long. Its plump body has 4 pairs of stubby legs, often ending in delicate claws. It has muscles, a digestive tract, a simple brain, and in many species, tiny eye spots. At the front are 2 sharp mouthparts called stylets. These pierce plant cells, algae, fungi, or small animals. A muscular throat then pumps out the contents. Some tardigrades even prey on other tardigrades. They are called water bears because of their lumbering walk, but the name hides something important. Every active tardigrade is fundamentally an aquatic animal. Even one living in dry-looking moss needs a microscopic film of water around its body. That film lets it move, feed, exchange gases, and reproduce. So a tardigrade in moss lives inside a world that can vanish in an afternoon. As sunlight warms the moss, its watery corridors shrink. An active tardigrade cannot simply store enough water and wait. Instead, drought-tolerant species begin a controlled collapse. The legs pull inward, the head retracts. Muscles shorten the body from front to back. The cuticle wrinkles into deep folds, and the animal contracts into a compact shape called a tun. Then it loses almost all its body water. This is not ordinary hibernation. A hibernating mammal remains wet, breathes, and runs a slow metabolism. A tardigrade in a dry tun reduces its metabolism until it becomes nearly undetectable. It does not walk, eat, grow, or reproduce. The animal has entered anhydrobiosis, a form of cryptobiosis triggered by water loss. That creates a huge problem. Water is not merely something inside a cell. It is the environment in which the cell's chemistry works. Proteins depend on water to hold their shapes. Membranes depend on it to maintain their structure. Remove water carelessly, and proteins can unfold or stick together. Membranes can leak, collapse, or fuse. Dissolved salts become dangerously concentrated. Reactive molecules can damage DNA. Drying an ordinary animal is less like emptying a bottle and more like removing the mortar from a building. The tardigrade has to preserve the building while nearly all the mortar disappears. Many organisms solve part of this problem using a sugar called trehalose. It can help stabilize dry cells and create a glass-like interior, but several well-studied tardigrades contain surprisingly little trehalose. They rely heavily on a different toolkit, including proteins found only in tardigrades. One important family is known as CAHS proteins. In a fully hydrated cell, these proteins are unusually flexible. Instead of folding into one rigid shape, they behave more like loose molecular threads. As water leaves the cell, everything inside becomes more concentrated. CAHS proteins move closer together and begin forming reversible fibers and gel-like networks. Imagine a soft internal scaffold appearing while the rest of the cell is shrinking. Experiments suggest this network can stiffen cells and restrict the movement of vulnerable molecules. In the dry state, CAHS proteins can also contribute to a glassy material that immobilizes cellular structures before they can collide, unfold, or merge. The cell is not protected by staying liquid. It is protected by becoming physically locked in place. In 2026, researchers tested one CAHS protein inside synthetic cell-like bubbles. Under drying stress, the protein changed structure, reinforced the model membranes, and helped preserve what was sealed inside. That does not mean CAHS proteins explain every tardigrade ability. Different species use different combinations of protectants, antioxidants, structural changes, and repair systems. Scientists are still working out exactly how these defenses cooperate inside the complete animal. But the visual idea is becoming clearer. As the tardigrade dries, its cells build temporary molecular supports. Then the whole system hardens into a stable state. When water returns, the process reverses. The ton absorbs moisture and expands. The legs emerge. The protective protein networks dissolve or reorganize. Metabolism restarts, and the tardigrade begins repairing whatever damage accumulated during drying and storage. It is not returned from death. It preserved the physical machinery required for life to resume. This distinction explains the famous survival stories and the myths built around them. Tardigrades really have survived direct exposure to space. In a 2007 experiment in low Earth orbit, dried tardigrades were exposed to space vacuum. Many recovered after returning to Earth and being rehydrated. Some also survived combinations of vacuum and solar radiation, although ultraviolet exposure caused much greater damage. That does not mean tardigrades can live in space. The animals in the experiment were dormant. They were not crawling, feeding, or reproducing in a vacuum. Space was something they endured for a limited time, not a habitat they could colonize. Radiation resistance is another real ability that often gets simplified. In one species, Milnesium tardigradum, researchers measured median lethal doses of several thousand grays of gamma radiation. That is astonishing, and the animals showed high resistance even while hydrated. But survival alone did not mean they were unharmed. Doses above 1,000 grays could leave survivors sterile. There was also no single radiation shield shared identically by every tardigrade. One famous protein, called Dsup, can bind to nucleosomes, the structures that package DNA, and help protect genetic material from highly reactive hydroxyl radicals. Other defenses include antioxidants and powerful DNA repair responses. Their radiation tolerance may partly be an accidental benefit of systems that originally evolved to handle drying, because desiccation also damages DNA and produces oxidative stress. Now for the biggest reality check. Heat can kill tardigrades surprisingly easily. In a study of Rhamazottius variornatus, active animals exposed for 24 hours had an estimated 50% mortality temperature of about 37 degrees Celsius. That is close to human body temperature. Dried tons performed much better, but exposure time mattered. Their estimated 50% mortality temperature was about 83 degrees Celsius after 1 hour, then fell to roughly 63 degrees after 24 hours. So when someone says a tardigrade can survive extreme heat, the missing questions are which species, in what state, at what humidity, and for how long. The same rule applies to freezing, pressure, chemicals, and radiation. A short laboratory exposure is not the same as permanent survival. A dry ton is not the same as an active animal. And one exceptional species does not represent every tardigrade. Even entering the ton is not guaranteed. Some species need time to prepare as their environment dries gradually. If water vanishes too quickly, they may fail to produce enough protectants or complete the necessary body changes. Different species, and even different populations, can have very different survival rates. They can also be crushed, digested, infected, or damaged beyond repair. Staying dry for longer generally reduces the chance of successful recovery. Claims of tardigrades reviving after more than a century are not considered strong evidence. One of the best documented long cases involved Antarctic tardigrades, recovered from material that had remained frozen for just over 30 years. That is still remarkable. It just is not immortality. A ton also cannot build a tardigrade population. It produces no offspring and gathers no energy. It is spending stored resources to preserve the possibility of a future. If liquid water never returns, the pause never becomes a rescue. And that reveals what cryptobiosis is actually for. Tardigrades did not evolve their strange biology to survive spacecraft, radiation machines, or physics experiments. Those are human-made tests that push an adaptation far beyond its everyday job. The real threat was probably much smaller and far more common. A film of water around a grain of soil disappears. A patch of moss dries between rainstorms. A temporary pond freezes or evaporates. For an animal smaller than a printed period, that is the end of the world. The tardigrade survives by transforming before the end arrives. It folds its body, removes the water that makes dangerous chemistry possible, braces its cells with reversible molecular scaffolds, and waits as a compact ton. The toughest animal is not invulnerable. In fact, while active, it can be surprisingly delicate. Its genius is knowing how to stop being active. A tardigrade does not defeat death by fighting through every extreme. It survives by becoming dry, still, and almost biologically silent until its tiny world exists again.